All right, this time let's talk about how did we figure out that DNA was a real thing. This did not happen all at once. In fact, it took many scientists decades of research to figure out what this DNA structure was. So let's dive into the annals of science history. First off was a scientist named Friedrich Miescher in 1869. He was the first one to isolate DNA. At the time he called it nuclein because it was found in the nucleus. He did not know its function, he did not know its structure, just that he knew it was made of carbon, nitrogen, oxygen, hydrogen, and phosphorus. But he was the first one to isolate DNA in cells and knew it was from the nucleus, hence the name nuclein. Pierre Levine he figured out, hey, there's 4 nitrogenous bases, that's adenine, thymine, guanine, cytosine, A, T, G, and C, and that there's a phosphate group and a sugar group. Again, he knew these were from nuclein, and by then I think it had become called DNA. But he didn't know how these things were arranged, just that, hey, these are the proportions of these chemicals in this macromolecule. Macromolecules are very large molecules. All right, but again, he didn't know it, wasn't quite sure on its purpose or how it was built, just that this thing is here, it is big, and these are the main groups within it. Chargaff, who mentioned last time, he's the one that noticed, hey, adenine and thymine are always found in equal amounts, as are cytosine and guanine. So this, Chargaff's rule is a really important clue for later scientists to figure out, hey, this is how it functions. So Chargaff's contribution was this simple equation that adenine and thymine are always in the same concentration as is cytosine and guanine. The first image of DNA is the work of Rosalind Franklin and the lab she was in. For a long time, she was not given proper credit. This is one of the controversies in science right now is what proper credit should she have been given. She was the first one to use X-ray crystallography to take this picture. That's picture 51, as it's called. They gave the first real look. Okay, what was the diffraction pattern? So it wasn't like a snapshot. It was a diffraction of how light bounced off this molecule. So you get the strange X-shaped patterns. The question became, what molecule would fit this structure? Unfortunately, Rosalind Franklin died young from cancer, likely caused by radioactive elements from her lab. And she was a difficult person to work with, as Watson and Crick found out. We'll talk about those guys next. But it's important to note, she was the first scientist to be able to take this X-ray crystallography picture of DNA, which is crucial for the next guys to figure out its structure. James Watson and Francis Crick were 2 rather brilliant scientists. I believe James Watson is still alive. He has a TED Talk. Look it up if you're curious. He's an interesting fellow. And they set up with a really strange idea against the current thought that maybe it's the nitrogen bases, A, T, G, and C, which make up the middle of the DNA molecule. However, they gained access to Rosalind Franklin's pictures without her exact permission and didn't really give her all the credit she deserved, hence in the controversy about these 2 fellows. Like, did they have the right to her research? Be that as it may, they were able to figure out which structure, which model of DNA would fit all these things that DNA needed to be. And turned out they stumbled on the right configuration that every piece of genetic science relies on, is this idea of the double helix spiraling about, sugar phosphates on the outside, with the new nitrogenous bases on the inside. 4 essential things about DNA that were so hard to figure out that Watson and Crick stumbled on based on Rosalind Franklin's work. You have the sugar phosphate backbone on the outside, the double helix shape, you can see it spinning in the corner. The strands run anti-parallel. We'll talk about what anti-parallel means in a few minutes. Basically means they run opposite directions to each other. We'll cover that in a bit. And that there's specific base pairing, A with T, G with C. These are the essential things with DNA we need to remember to understand its structure. And this is what undergirds all of really biology right now, is this genetic knowledge. So it was a huge breakthrough. It took over a century to work it all out. And it is now the basis of biology and medicine and so many other things that it's profoundly changed our understanding of life and evolution. And it's countless things. It's hard to say everything that it has impacted. But again, these are the 4 essentials. So remember these and you'll be at least have a good starting point. All right, that is it for this lesson. Next time we'll talk about some more genetics and about how that information is passed on to the cell in order for the cell to know what to do.